So in the last lecture, we have seen the part 1 of the revision. Today we are going to see the part 2 of revision. As we have already seen in previous lectures, we are just going to revise it today as the revision part 2. In today's lecture, we'll be revisiting the fundamentals of phylogenetic trees. If you remember it, its construction and analysis using BioPython. I know you all already know what BioPython library is. We have already laid the foundation in our earlier sessions, exploring the basics of BioPython and the bioinformatics. Now it's time to refresh our understanding and delve deeper into this fascinating field. Now, before we proceed, let's recap what we have learned so far. BioPython, as many of you know, is a versatile toolkit that simplifies complex biological data analysis. It allows us to manipulate and explore DNA, RNA, single strands, and protein sequences with ease. In our previous videos, we covered the basics of BioPython, how to read and write sequences, access sequences information, perform simple analysis. Today we are going to, also we have seen some applications of those skills to construct and interpret phylogenetic tree. Now the codes for this also I have kind of provided, and now let's revisit some fascinating bioinformatics that we have already seen. We'll start by importing our trustworthy BioPython module. You already know it. Then read sequences from various species. We'll give them a meaningful ID for clarity and create a multiple sequence alignment to compare their similarities. So we'll kind of judge, compare, we'll kind of see the similarities between them. Next, we kind of calculate the distance matrices. So this is the place we are calculating distance, whatever distance that we are getting, we are kind of taking it in the matrix. It is a vital tool for measuring the genetic differences between our species. This matrix, I hope you can see this. This matrix will serve as a foundation for the phylogenetic tree, as you all already know. With our distance matrix in hand, we have already seen this distance matrices. So with our distance matrices in hand, we'll construct our first tree using UPGMA algorithm. I hope you are not hearing this for the first time. UPGMA algorithm. Now the tree will represent the hierarchical relationship between the species based on their genetic similarity. We won't stop there. We'll explore our tree, find common ancestors, and discover how interconnected this trees of life truly is. Then we'll briefly touch on neighbor joining algorithm. So what we have seen earlier, UPGMA algorithm. Next, what we saw was neighbor joining algorithm, especially for the tree construction. By the end of the lecture, we also refreshed your understanding of how to construct, visualize, and analyze phylogenetic trees using BioPython. So let's now try to recap it. I hope you all are ready for this. Fantastic. Let's dive back into the intricate of integrate word of this phylogenetic and biopython. Strap in, folks, it's going to be a fascinating ride. So we have seen the codes, we have seen the snippets. In the complete code and snippets, we kind of drew the tree. That kind of, in the conclusion, we can say that our revision session on the phylogenetic tree constructions and analysis with biopython kind of refreshed our knowledge, and in the upcoming, like this particular technique, we will see other forms of exploration that we have already done. I don't think there is much problem with this particular tree construction. Any which ways, if you have any problem with the construction of tree or any of the doubts related to the codes, you can of course anytime ask in the portal. This is open for you. Now, as we continued our journey into the Python, we also see another thing in the previous videos, that was ordinary differential equation. We generally called it ODEs. And the hilarious world of biology, I hope you all remember, we embarked on a scientific comedy of epic proportions. Tried to make it as hilarious as possible. I'm bad at creating jokes, but I tried. The Python from lab codes to the laughs, a biologist's comedy of ODE, we tried to create it. And because ODE is actually a very difficult thing to teach. So in the first part of our comedy act, we unrevealed the secrets of modeling exponential growth and population dynamics, especially using Python. But that was just the beginning. That was just beginning of this adventure. The next thing we represented the kind of case study which says Python, the contagious comedy, the disease is spreading extravaganza. We got ready to dive into a zany world of Python powered ODEs as we demystified the secret of disease transmissions. I hope you are remembering whatever I'm saying. You are all remembering the codes behind it and the things behind it. It's just what this is just a recap of what we have already done. Before we jumped into the hilarity of modeling disease spread, we made sure Python was our partner in crime. Now, setting up the outrageously hilarious informant as we did it, just as we did our first act, we rushed into the python.org. Of course, downloading different things. If you don't remember it, I'll again do a revision for you. Don't worry. Python download, we just download the Anaconda to ensure the Python we have. Next, we summon the essential libraries by executing the magical command, that is pip install, and we say pip install numpy, scipy, matplotlib, whatever we want to install. The ODE that kind of tried to made a hilarious act on this. To model disease spread, we presented the legendary SIR model, susceptible, infectious, recovered model, if you remember it. We beheld the ODE that left us in stress. Now, we got ready for a laugh riot as we set up the initial conditions for susceptible, infectious, and recovered individuals. We kind of time travel for 200 days. We took S naught, I naught, R naught, and time points. I hope you are remembering them all. Finally, after doing all things, we used scipy's ODE function to solve the SIR model, the model like using ODE, and then we kind of witnessed the disease spread extravaganza. We gathered our audience and created a jaw-dropping plot. Now, after plotting everything, we had mastered the art of modeling disease spread. We let our creativity run wild. We adjusted the parameters, threw in some plot twist, and explored the world of disease control with Python as our trusty sidekick. And now, for a surprise comedy act that biology threw our way, we imagine modeling a population of narcoleptic kangaroos, if you remember, with an affinity for disco dancing. I know it's all bad jokes. Python's ODEs were here to help us bring this absurdity to life. In this unexpected twist, Python and ODEs were about to take us on a dance floor adventures like no other. We got ready to unleash the kangaroo party. We set our initial conditions and the time spent for this disco inferno. I hope you are kind of remembering this. Python was our dance partner once again. We used SciPy's ODN function again to make those kangaroo boogie like there was no tomorrow. And finally, after doing this whole thing, we went on for another piece of code that kind of threw the disco fever plot when we prepared for the most epic kangaroo disco party visualization ever witnessed. And after going into this all things, in the end, with this all hilarious type of teaching that tried to make ODE very simple, we had officially joined ranks of biology's funniest comedians with Python as our comedic sidekick. We could model the complete scenario for dancing kangaroos to roller coaster population. We dive deeper into the world of biological ODEs, explore new frontiers, and let our imagination run wild. In the end, we remembered that Python was our partner in the scientific mischief. The world of biology was full of surprises. And with the Python, we were equipped to tackle them with a humor and curiosity. We kept coding, we kept laughing, we kept exploring in this fascinating universe of biology. After this, as we continued our journey in the realm of Python, ordinary differential equations, the ODEs, and the hilarious world of biology, we embarked on a scientific comedy act of different proportions. So, for this, we kind of saw different things in which we saw from lab coats to laugh, the biologist's comedy act. In the first part of our comedy act, we unrevealed the secret of modeling exponential growth and dance with disco loving kangaroos. As I've already told this thing, that this is something that we done. But now, after doing this, in the next, we dived into a serious and impactful application of biology, and that was contagious disease spreading thing. In the next part, this contagious disease spreading thing, we kind of ventured into the realm of disease modeling and vaccination strategies using ODEs. For this all things that we have done, if you have just coded it in a right way, you will see that you are altogether complete different person or different biologist now. You know how to code, you know how to run things in a Jupyter notebook. That is still a lots of biologists who don't know. We explored the challenges of predicting disease trajectories and optimizing vaccination strategies. all with the help of Python and ODEs, the real life ODE application, modeling drug doses in chemotherapy we have seen. And as we revisited our previous lectures, we focused on the real world application, modeling drug doses, especially with related to the chemotherapy that we have seen, the cancer treatments in a delicate balance, and ODEs provide the mathematical tools to find optimal doses. So we cannot have a dose which is very high, we cannot have a dose which is very low. We need to find something in between that we try to do, and that is what we did with the use of ODEs, the optimal doses. We use the logistic growth equation combined with the drug effect term to model cancer cell growth and the drug impact. After doing this all things with the use of ODE in our experiment, in this experiment, we initialize the cancer cell population. We define the time points and set an initial low drug doses. You all remember it, right? Why not? The time points, the drug doses. We also said that the Python has some healing power. So Python was our best companion in finding this optimal drug doses. We iterated through different doses, simulated cancer cell growth, and measured the final population. I hope you're remembering everything. After doing this all, we kind of saw and we kind of succeeded by using ODE and Python. We found the optimal drug doses, which you also have the power now to twerk or to change according to you and find the optimal doses according to your real-time use cases. That minimizes the final cancer cell population. That's such a big thing that you have learned. Our exploration is in biotechnology and medicine doesn't end here. With Python by our side, we continued the complete exploration in the new frontiers and pushed the boundaries of the science. We have also seen the modeling of immune response systems. Remember, after we succeeded in the optimizing chemotherapy doses, we kind of use this ODE equation and its far-reaching impact that was related to a situation where a new infectious disease is spreading, and how you can predict its trajectory and assess the effectiveness of the vaccine strategies. For example, you have seen the COVID-19, so how we can actually see how it's spreading and how we can assess the effectiveness of the vaccine strategies that is related to COVID-19. So ODs offered a mathematical lens to examine all these things. We use simple SIR model again for this, which is susceptible, infectious, recovered. The dynamics of an infectious disease is much like one we explored earlier. So we have done this SIR model even in the previous lectures, even in this lectures, even few lectures before this. So this is kind of novel thing that we tried to do. I don't know whether you liked it or not. In this scenario, we initialize the population conditions, define time points, specified disease parameters of our experiments, which was, if you remember, we used S0, we used I0, we used R0, we used time points, beta, gamma, if you remember this. Again, Python saved us from doing the complex mathematical calculations that we in general need to do. Python came for our rescue kind of. We used SciPy's ODN function to simulate the spread of disease and assess various vaccination strategies. In the end, it was triumph of the science. By using ODEs and Python, we modeled the spread of infectious disease, access the vaccination strategies, make some informed decisions to protect the public health. We kind of won this battle. So one of the again new field is machine learning, wherein it can assist in experimental design by helping biologists optimize their experiments and sample sizes based on data driven insights. This ensures that the resources are used effectively and experiments are designed to yield meaningful results. Why I'm telling this all? Because in the end we need to know what exactly this computer science, the field of machine learning, artificial intelligence, data science is helping you out in doing things. It is also kind of helping you in real time monitoring. So in the field like ecology and environmental science, real time monitoring of data or the data is crucial for tracking changes and responding to emerging challenges. Machine learning can provide continuous it actually machine learning can provide continuous analysis of sensor data, enabling timely interventions in case of ecological disturbances or environmental threats. Next, we can also see a scientific innovation. For example, by embracing machine learning, biologists have the potential to drive scientific innovations beyond traditional boundaries. As we have already seen it, they can explore some very new research, create collaborations, and can have groundbreaking new experiments. In the face of global health, like again the same thing, like infectious disease outbreak, machine learning can play actually a very crucial role, especially in terms of in the times of pandemic. I have already told you this. In the contact tracing, the forecasting of spread of diseases, biologists who are proficient in these techniques can contribute in the public health and especially in the domain of crisis management. So many bioinformatic tools and software packages are built upon the machine learning algorithms. Biologists who understand machine learning, actually they are going to be they're not going to stop, and they would be the pioneer of their research in the biology. Machine learning is a field that continuously evolves with new techniques and algorithms which are emerging regularly. In fact, these times even every day a new paper is coming and which is blowing our mind. ChatGPT 4, some stable diffusion, things like this. So biologists who invest in learning, machine learning, deep learning, AI can adapt to these changes more rapidly and stay updated with the latest advancement in this all fields. Machine learning promotes the principle to open science by encouraging data sharing, the code transparency, as I've already given all the codes to you, and collaborations. Biologists who embrace these practices contribute to more open and collaborative research environment, fostering the sharing of knowledge and accelerating scientific progress. Before going to one more method, which is solving biological complex data, let me tell you that there might be some biological terms that I may be taking them wrong. Please forgive me for that. But machine learning can provide innovative ways to approach these mysteries, offering fresh perspective and methodologies for unrevealing secrets of lives and nature. Again, please forgive me if I'm pronouncing any of the biological terms incorrectly because I'm not a biologist. In summary, machine learning is no longer an optional skill for biologists. It has become an essential tool for advancing research, solving real world challenges, and driving scientific discovery in the 21st century. Biologists who embrace machine learning not only enhance their own research capabilities but also contribute to the broader scientific community's collective efforts to address critical issues in biology, healthcare, and environment. It is valuable investment in the future of biology and its potential to impact the world positively. So, ladies and gentlemen, as we draw the curtains on this captivating journey through the world of Python, ordinary differential equations, the different graphs, different things, biology, and the boundless wonders of science, I want to leave you with resounding message of inspiration. Throughout this adventure, you have witnessed the incredible power of knowledge, the magic of mathematics, the limitless potential of Python. You have laughed, you have learned, and discovered the science is not just a collection of facts, but a grand narrative filled with curiosity, exploration, and the audacity to dream. As you embark on your own scientific odyssey, remember that the pursuit of knowledge is a path paved with curiosity and resilience. The challenges will come, the challenges will come, equation will confound, and the experiments may fail. But in those moments, you will find the true essence of discovery. For it is in the face of adversity that your brilliance shines brightest, and your determination fuels the flames of innovation. Bella ciao. My fellow seekers of wisdom, for it is a song of freedom and resilience, echoing the spirit of those who fought for the better world. Just as they march forward, undeterred by the obstacles, so too shall you forge ahead in your pursuit of scientific excellence. The world eagerly awaits your contribution, your breakthroughs, your laughter in the face of complexity. You are the torch bearers of progress, the dreamers of the brighter future, and the architects of the change. With Python as your ally and ODE as your muse, there are no limits to what you can achieve. So, my dear students, as you venture forth into the unknown, remember to keep your curiosity aflame, your dreams boundless, and your determination unyielding. Bella ciao. And may your scientific journey be filled with wonder, purpose, and the resounding echoes of laughter in the pursuit of knowledge. The world is yours to explore, and its mysteries are waiting for you to unreveal. Farewell, and may your path be illuminated by the brilliance of your ideas and the beauty of your discoveries. Until we meet again, keep coding, keep experimenting, keep changing the world. Bella ciao, my friends. Goodbye.